Our next speaker, which we're very excited to hear from, Anders Nykjaer, Professor of Medical Biochemistry, Director of the Lundbeck Foundation Research Center, the Danish Research Institute of Translational Neuroscience, who will talk about SCORE-CS2 regulates dopaminergic wiring and BDNF dependent plasticity, and a little something extra at the end.
Oh yeah, that's right. I'll present for you. Can you hear me? I'm not sure. Okay. Yeah, it's right. My talk will be followed just by 1 or 2 minutes introduction to the brain prize. So this was the original title of my talk, but in the sake of time, I have decided to cut away this part. But I'll be happy to discuss it with you afterwards if anybody's interested. Almost 20 years ago, before the human genome was known, we by biochemical means purified a family of receptors that we called the VPS10P domain receptor family, or sortilins. And the VPS10P domain is shown here in green. That initially was found in yeast, where it's a sorting protein that targets protein to the vacuole. The receptors are enriched in the nervous system, although not entirely expressed. It's also found outside in highly specialized cells, such as in the pancreas and also in hepatocytes. The biology of the receptors is quite complicated, and is also the cell biology, because in essence the receptor can be everywhere, or they can. Not surprisingly, they follow through via the trans-Golgi network to the plasma membrane, but from here they can be internalized, and not only will they go into recycling endosomes, but a pretty large part of them will go back to the trans-Golgi network, depending on what kind of cytosolic adapters they bind. However, they can also do direct sorting from the trans-Golgi network into early endosomes, and that may be a way to deliver lysosomal enzymes. Recently, it was also shown that they can actually be present in secretory vesicles or dense core vesicles, and now we know it also exists in a soluble protein. So it has been a very, very hard travel to figure out what are they actually doing. Luckily, human genetics has told us a lot, and also studies in transgenic mouse models has validated some of these associations. Like, for instance, it's important to neurological disorders like multiple sclerosis, frontotemporal lobe dementia, but also in mental disorders like bipolar disorders and schizophrenia. Cognitive dysfunction is also associated with the receptor, and perhaps not surprisingly, considering that some of the receptors exist only also in the beta cells in the pancreas with metabolic disorders like type 2 diabetes. I'll focus just as a background a little bit about sortilin, because that was the first we figured out what was doing, and it's a very important function in regulating neurotrophic activity. Neurotrophins normally target a tyrosine receptor kinase, and there are several neurotrophins: NGF, BDNF, NT3, and NT4. They're essential for development of the nervous system. It's also important for the maintenance of the integrity of the adult nervous system, and they have been engaged in plasticity as well. There are 3 receptors. NGF binds track A, BDNF track B, NT3 track C, and then we have NT4 that targets, in particular, track B. Now, there's yet another receptor, that's a promiscuous one called P75, because it can bind all of them. And what we now know is that it's kind of a bystander that increases the affinity and specificity of the TRK receptors towards a cognate ligand. In 2001, Barbara Hemstedt showed that NT was produced in the pro form, perhaps not surprisingly similar to proinsulin that's converted into insulin. But what she found was that this molecule has a function in its own right. Because it's apoptotic, induces cell death. And it does so in a way that requires P75. We found that not only P75 is required, but also sortilin, because it captures the pro-NT together with P75. This means if you don't have sortilin expression, the pro-NT would be converted into a mature counterpart. It would bind the TRK receptors, P75 will be recruited by the TRK receptors, and we'll get trophic support. Now, as you can see, P75 has two functions. It's apoptotic, but in another context, it can also be trophic. And to our surprise, it seems to be this case also for sortilin, because it can target TRK to the plasma membrane. So in the absence of sortilin, you have a less robust signaling by neurotrophins. And one way of demonstrating this was that we crossed P75 knockouts with sortilin knockouts, hoping that would basically tilt everything to make the animals sick. And this is actually what happens. So here we have the peripheral nerve, the REMAC bundles in 3-month-old animals, and those are nociceptive fibers. And in the P75 wild type and a sortilin knockout, were pretty much normal, but they're completely degenerated and double knockout. And as a consequence, you can see they had severe deformity in their hind limbs because they couldn't feel the pain. They didn't know where they were basically walking or climbing in the cages. So it was kind of a prize for autonomous behavior. Now, what we know is that sortilin targets the TRK receptors to the plasma membrane along the axon and also along neurites. So it's absent. There's still transport, but it's less efficient. Whoops. I'll talk a little bit now about SOX2. And SOX2, because it's similar to sortilin structurally, it's also a sorting receptor. We asked, could it be that SOX2 is also involved in TRK signaling? And why we did so was there were a lot of human genetic data coming up at that time that suggested SOX2 could be very, very important in plasticity, as it's genetically linked to both bipolar disorders, schizophrenia, and also ADHD, and that has now been recapitulated in several other studies. It's not present during development, but it comes up at P30 in the hippocampus when synaptogenesis is really taking place, which also supports a function in plasticity. We look where it's expressed, and it co-localizes with neuron markers. It's never in glial cells, and it's present in in dendrites. It never co-localizes with the axonal marker in NF200. So we took out the brain, we did a subcellular fractionation to figure out, is it localized in the presynaptic site, which you would probably not predict at what we've seen here, or is it at the postsynaptic site? And actually, it was enriched in fractions that contained the marker for postsynaptic densities, whereas it was absent from the presynaptic fractions. So TRK-B and BDNF are important for plasticity. TRK-B is present both on the presynaptic side and the postsynaptic side. Here it can bind ligand BDNF, and it may facilitate release of glutamate, but it may also facilitate activity of NMDA receptors and AMPA receptors. It does so by activating specific pathways, either directly to the NMDA receptors here, or it can activate it indirectly by the phospholipase gamma pathway that stimulates this sodium calcium, what is it called, channel, and would change the membrane potential and increase probability of opening of the NMDA receptors. We did not have a presynaptic phenotype, which we'd also expect to be normal since SOX2 was not in the presynapse, and this is just classical paired pulse facilitations. So we were interested in the postsynaptic sites. What's going on here? And these are field recordings, classical field recordings from the CA3, CA1 synapse. And what you find is that in the wild type, you can maintain LTP, which is called the late phase LTP. Obviously, you do have the early phase, that's the first 30 minutes, but it's maintained, which is not the case for the SOX2 knockouts. This was not a developmental defect, because if we took a wild type and added antibodies against SOX2, we found exactly the same. Surprisingly, we could, if our model was correct, we could rescue this phenotype by adding into the perfusion medium high concentrations of BDNF. However, it seemed that sensitivity towards BDNF, after all, was reduced because we could only rescue when we used very high concentrations of BDNF. Here we took a BDNF heterozygous mouse that obviously have only 50% of its normal concentration of BDNF, and that also fails to induce late phase LTP. However, if you add in BDNF, you can see around half a nanogram is sufficient to give half maximal LTP. In the SOX2 knockout, you needed up to 10 times as much. And we did find that these neurons were less responsive to BDNF. Here we have, those are hippocampal neurons that were cultured, and we added in BDNF. And it's very clear that there's less autophosphorylation of TrkB and also total phosphorylation. Those are wild types with increasing concentration of for BDNF, and here are the knockouts. Also, phosphoACT could be robustly induced in the wild types by BDNF, but not so in the knockouts. There was also a difference in PLC gamma. So altogether, this suggested that they do respond to BDNF when present at very high concentrations, but they are certainly less sensitive. If this was the case, we should be able to culture neurons and also show they do not respond as well in terms of branching and spine density. And this is what you see here. Here you have wild-type neurons, and you can study the number of branches at the first, the second, and the third order in the absence or in the presence of BDNF. And BDNF induced more than the doubling in the number of branches in the wild type. However, they were completely unresponsive in the knockouts. The same also applied to the number of spines. They responded beautifully to the wild type by increasing the spine number by almost twofold, whereas we saw no effect in the SOX2 knockouts. Well, what is going on here? We asked if SOX2, very much similar to sortilin, could bind directly to TrkB receptors. And this is a culture of primary neurons. And we did a pull down for SOX2, and you can see TrkB came along, but it didn't do so in the negative control. To get a more spatial information about where is SOX2, where does it bind TrkB, we took advantage of the proximity ligation assay, where you can visualize two proteins interacting. Here it's PSD95 and TrkB that would appear red, by having two antibodies that are coupled by circular DNA probes. Once they're getting proximity, they can be visualized by using fluorescently labeled nucleotides. So here you have PSD and TrkB in proximity, and this is then a double staining for SOX2. SOX2 is along neurites, but it also gives these yellow spots, strongly suggestive that all three components are present once in a while in postsynaptic densities. So what does SOX2 actually do with TrkB? Again, we used the proximity ligation experiments, but now we used SOX2 and TrkB. So whenever you have a red spot, that shows you that TrkB and SOX2 are in proximity. There is, or normally, if you have a culture, there will always be some kind of activity going on in those cultures, and you can certainly see also they do interact. If we stimulate with low frequency, that does not induce long-term potentiation, but in depression, they seem to go away from each other. But if we induce potentiation, they come together very, very strongly. We then ask, does this impact on how much TrkB we have in postsynaptic densities? Now we used a probe against PSD95 and TrkB. And again, we saw a robust targeting of TrkB to postsynaptic densities. This may not be surprising, that has been published before, but when we did the same in the SOX2 knockout, TrkB never ended up in postsynaptic densities. That we can quantify. We can use imaging and of course, and here you have the controls, how much co-localized of SOX2 and TrkB in the absence of stimulation, and then in presence high frequency, and it does go up with almost threefold, and it comes apart when you try to induce long-term depression. TrkB was also targeted, as I showed you before in the pictures. If we stimulated with high frequency, it went to postsynaptic densities, whereas there were no effect on low frequency stimulation. No effect was observed in the SOX2 knockouts. So this indicates that SOX2 is very important for targeting TrkB in an activity-dependent manner to postsynaptic densities for synaptic transmission. We wanted to show this using the two-pathway stimulation analysis, and this is based on the synaptic tagging hypothesis that was proposed or initially put forward by Richard Morris. And the thing here is, the idea is if you stimulate one ensemble of neurons with high frequency, that would robustly introduce synapse formation on consolidation and long-term memory. This is what you can see here. But it requires two events. The first thing is that that the area that's going to develop in or be strengthened into a spine needs to capture certain molecules called tags. At the same time, you would need to have strong expression of proteins called plasticity-related proteins that have to bind either from the outside or the inside to those tagged molecules, and this is absolutely critical for synaptic consolidation. So Bailou and colleague, he suggested actually tract B could be a synaptic tag in the sense that if you would stimulate the synapse, it would go here and then it should bind a BDNF that's also being released upon active or strong synaptic stimulation, high frequency. If you now have a neighboring neuron that is weakly stimulated, that can induce targeting of the tag of the tract B to the tag here, but it fails to induce plasticity-related protein synthesis. So that means that in order for this one to induce robust LTP and memory, it has to capture some of the BDNF that's produced as a consequence of the high frequency stimulation. So we took advantage of this to demonstrate that SOX2 is indeed important for the targeting of tract B to these tags. So if we stimulate with high frequency, that will induce LTP. Low frequency will not, unless it's able to capture BDNF. So if we did this experiment here in the presence of antibody against SOX2, we would predict we should fail to induce LTP when we stimulate it with low frequency in an independent population of neurons. First, we used two stimulation electrodes, one both in the Schaffer collaterals but activating different pathways neurons. So if you stimulate here with high frequency and you stimulate here also with high frequency but with lower intensity, for both situations we record in the same neurons. And we wanted to show first that these two pathways are independent. So if you stimulate at S2 alone, it induces EPSP, but it's completely unaffected whether you stimulate S1 first. The opposite also holds true. So we have two independent pathways that converge on the same cells where we do our recordings. Now, if we stimulate with low frequency, I said it's unable to induce LTP, and this is also what you see here. You can get the early phase, but it declines back to baseline. If you stimulate with high frequency, it remains as you would predict. If you first stimulate at S1 with high frequency and then the low frequency at the other side, S2, all of a sudden you can maintain late phase LTP, which is required for synaptic consolidation. But how do we prove SOX2 is involved in this? Well, we did the same experiment again, high frequency stimulation. But if we then added in antibodies against SOX2, now low intensity, high frequency stimulation failed to maintain LTP. Control antibodies did not. So SOX2 is required for targeting tract B or capturing tract B and transport it into what's going to develop into that's going to develop into the consolidated spine. This can be studied also in what's called the behavioral tacking paradigm. And what you do here is that you have mice and you train them, you give them very mild shock, and you study the time it takes them to go. First you put them into a light room, and then they would like to go into a dark room, but once they enter, they will get a mild shock. Both of them learned this or did this equally well during the training period. And after 1 hour, if we retested them, both of them remembered. But in order to get long-term memory, you need to have either high stimulus or you need to get your to see something else. You need to observe another thing that can make sure you can convert your LTP, the weak LTP into a strong LTP and consolidate your memory. And after 24 hours, none of them remembered because the stimulus were very weak. If, however, we expose the animal to novel objects 15 minutes before the experiment, so that should mimic what you see when you do the synaptic tacking. You stimulate with the object recognition of the visual input. The wild type, all of a sudden, they could remember the shock 24 hours, but the SOX2 knockouts could not. We could also test them in the elevated plus maze. I'll do this very fast. You know, you put a mouse in here, it prefers the closed arm, but once in a while it will enter the open arms because of curiosity and its activity, hyperactivity, or risk-taking behavior, depending on how much it does it. And here you have the distance. The knockout seems to walk a little bit more, run around, so they were hyperactive. They also entered the open arms more, and they stayed there for longer, showing that they were more risk-taking. So this is a kind of a feature of manic behavior. And 2 more slides, if we skip that one. And finally, we could study them in what's called the Prepulse Inhibition. This is a test that's kind of a diagnostic for schizophrenia. And that is also used actually in humans. So what you do is you subject the animals to a sound from 0 and up to 100, or let's say 80 and up to 120 decibel. And if you hear such a sound, you'll startle, right? You'll get this reflex. But if you hear one first, exposed to one at 80, then if you get a second one around 120, you don't react to the same extent because you're somehow prepared. If you have schizophrenia, you don't have this inhibition, or this prepulse inhibition. This is what we saw here. The SOX2 knockout failed to respond to the first tone by inhibition. This is a function of sortilin, long-range transport. But what does SOX2 do? Well, it acts here in the synapse. So once the TRAC receptors that come up into the neurites, SOX2 is taking them out in an activity-dependent manner to tag the synapse and consolidate the synapse to secure long-term memory formation. And by this, I would like to acknowledge the people here. I don't know why some are in bold and some are not. It doesn't mean they're more important than the other ones. The first part of the study on sortilin and anterograde transport was carried out by Christian Vector. The second on synaptic plasticity and SOX2 was a study shared by Ulrich Bölkow and Simon Gleo. There have been quite some collaborators, in particular Thomas Wilner, Moses Chow, and Barbara Hempstead. And you may notice that it says Lundbeck Foundation and Lundbeck Foundation, but what is a Lundbeck Foundation? The Lundbeck Foundation is very, very important in Denmark. It has 3 or 2 arms. One is the business. They have a substantial interest of the shares in the pharmaceutical company Lundbeck. And some of the revenues that they get from Lundbeck, they put into research, into free research, in particular neuroscience. So they give out every year around, and you have now to consider the size of Denmark, we have 5 million inhabitants, they give to neuroscience about $60 million. In addition to these 2 activities, they are also supporting biotech companies. But based on a major endowment from the Lundbeck Foundation, 6 years ago, they were established the Brain Prize, which was intended, and it still is, of course, to acknowledge groundbreaking research in neuroscience, and in particular of European neuroscientists. And when I say European neuroscientists, that means you at least need to have some strong interaction with your European neuroscientists. Or you have been born in Europe and you moved to the US, or if you're American, that you have strongly interactions with European scientists that will get the prize. So there are quite some possibilities. Now, last year, this prize was awarded to Graham Collinridge, Richard Morris, and Timothy Blish. And they got the prize actually for memory formation and synaptic tagging and long-term potentiation. And I know we have a speaker coming on Thursday, and that's Karel Svoboda. He got the prize last year. And it's a personal prize of €1 million. So it's not really that bad. Of course, there's some obligations coming along. One is outreach activities, and there are quite a few, I would say. And one of these is to come to Denmark here and for a meeting that we call the Hinskov meeting, and that's taking place in 2 weeks in the northern, in a small island in Denmark called Fralin. It's a very beautiful place and a wonderful meeting, and I strongly encourage you to come. It's not too late yet to sign up for it, and we have some very, very good speakers. And finally, I would also, at this occasion, like to encourage you to nominate candidates. It's too late this year. The next nominations or calls for nominations will take place in May, and the deadline is going to be September next year. But please come to Denmark if you want. I'm sure it will be a very, very nice and productive meeting.